God damn, Jimmy. This some serious gourmet Me and Vincent would have been satisfied with some freeze-dried Taster's Choice, right?
Welcome to Coffee Literature Review. I'm Chris Hendon. I'm joined by Doran Pennington. Today we're reviewing a paper published in the journal Food Chemistry in 2023 titled Pouring Hot Water Through Drip Bags Releases Thousands of Microplastics into Coffee. Joining me to discuss this paper is science communicating professional Alex Dainis. I've known Alex for a little bit. She's a film producer, works with the Dreyfus Foundation where we get some of our funding. She's one of the best science communicators in the country, PhD in genetics from Stanford. We're really lucky to have her. Welcome, Alex. I was going to ask you that because today we're recording right after the US election, and whether or not you're happy with the outcome, there's typically an impact on science funding depending on which party gets into power. And that can go both ways. I mean, historically, it's flip-flopped as to who supports certain arms of the federal research. Sometimes the Republicans last time cut EPA money. You sometimes see NASA money get cut for different reasons. So it just moves around. My question to you is, what are you most concerned about coming into the next 4 years for science in America?
I think one of the biggest things that I'm concerned about is, yes, funding, but the root of that being growing distrust from the public. Because we have seen, and there's a great Pew Research study that's been quoted 1,000 times, that the public's trust in scientists to do the right thing is dropping. And it's dropping across political parties, but it is dropping fastest among people who identify as Republicans and conservatives. Mm-hmm. And I am quite worried based on things that Trump has said about putting someone like RFK in charge of big swaths of health and science in his government. That is something he has said he will do, whether he means it or not, nobody knows. But that is someone who has been adamantly anti-vaccine, who is anti-fluoride, who is an HIV denier. Putting someone like that in a position of power in a place where the public already is losing trust in science, I think could do irreparable harm to how people think about science and how people trust science. And then from that, how they vote, how they want to fund science and where it goes from here.
So yesterday I was on Twitter, as many of us were, and I saw Taylor Sparks, professor at University of Utah, host of the Materialism Pod, post a tweet that says, "I'm genuinely excited to vote for the future without censorship, government waste, wars, and avoidable chronic disease. Hashtag Trump 2024. Following along, personally, I was on board with like every single word he was saying up to the point where he mentioned avoidable chronic disease and then, of course, the hashtag. And I challenged him on it, politely challenged him on it. And you can read this, it's on Twitter. And his response was that diabetes and obesity are avoidable chronic disease and that he's supporting RFK because he wants RFK to address the health problems in the United States. This is really a head scratcher for me because we're also talking about another highly educated person here who has the same data set and is concluding the exact opposite to you and I, which means that we don't only have an external problem with science communication, we also may have an internal problem with science communication.
Yes, 100%. This is a soapbox that I will get on loudly for as long as anyone will let me. I think we absolutely have a problem with scientists being able to communicate to other scientists, not even just outside of their field, but outside of their lab. I think we get so focused on our own specific little corner of science that we lose the ability to talk to people across the hall, never mind somebody, outside of academia. And I think that this is a problem that people are trying to solve, and there is definitely a wave of people who are trying to get communication to be a bigger part of this. Having been in science communication for a while, have seen the phenomenon that happens all the time, and I'm sure you see it all the time with coffee, that you get one study that says 3.5 cups of coffee a day is exactly the right amount of coffee to keep you from getting diabetes. And a study comes out the next day that says 3.5 cups of coffee a day is exactly the right amount to give you diabetes, right? And and I don't know why, actually. And I wonder if you do know why it is so prevalent in food science. I think it's because it's a good headline. I think it's because it's a catchy headline that the media can latch onto, that people like food. And so we can make food science headlines go big. but just this back and forth and nitpicking over teeny tiny little papers that then rolls itself into these big movements against different types of food. I took out some of my election anxiety yesterday by very kindly, uh, bringing fact-based information to anti-vax spaces online
Oh, that's
because
what
that
you
was
did to
something
release?
that's what I did yesterday, yes. And somehow wound up in these spaces on the conversation of vitamin C, which is mostly kind of a scam.
Yeah.
Vitamin C kind of does nothing for your cold, but there was one study that Linus Pauling really latched onto and it got a great marketing job and it went everywhere. And I could go on forever, but
Yeah, this
I just think there's a lot happening here and it's very scary.
this concept of trust in science is interesting because, the type of research that I do for my PhD, I'm doing electronic structure of porous materials. I have nothing to gain by, lying to the public about this. No one really cares outside of this niche of academics. But I think if you're operating in food science, in public health, in things that impact people's daily lives, you do have a really big responsibility to be, as thorough as you can be with the design of your research and with how you communicate it. And I mean, we've seen just in the coffee lab the way that news agencies will sensationalize the articles that we put out to make it seem like we're saying something that we're not. I mean, how many people that hear this are actually going and reading the paper and even just looking at the abstract? I mean, it's there's such a problem of the gaps of communication from scientists communicating to other scientists, scientists communicating to news outlets communicating to the public. I mean, there's a lot of places where things can go wrong.
Yeah. Alex and I were talking about this maybe a couple weeks ago about how not everybody should feel compelled to be the outreach person for a particular field. And not everybody's skill set is necessarily conducive to being always the lead author on a paper. It's like, a lot of different ways to partake in science, but there is really important to have good people being the quarterback looking downfield at all the other people, right? That's not the same. That doesn't mean they're better or worse. It just means that skill set someone needs to take that job and do a good job of it. That's the number one thing. Alex, you're good at it. I've told you this before, but you're really good at it. I'm about to say two words I've never said in my entire life to you, okay? Hypertrophic cardiomyopathy.
Hypertrophic cardiomyopathy.
All
You were
right.
close. You were really close.
He's Australian.
All right.
It's a regional.
Yeah.
All right,
Yeah, I'm sure.
Yeah, that's how we say it down. No. So why I'm mentioning this, of course, for the audience is that I do need to give some sort of introduction for Alex. You can tell that we go back a little bit, but from an academic perspective, Alex, you were an undergrad at Brandeis University, which is in the outer suburbs of Boston, and you did an unusual degree, right? It was what, biology and film double major?
Yes, exactly. So technically it was film, television, and interactive media studies, which is the longest name ever. But yeah, I couldn't decide what I wanted to do, double majored in both, and now, gosh, I don't want to think about it. 13 years on, I'm actually professionally doing both, so I'll take it.
Yeah, you're vintage now. But then you did a PhD in genetics at Stanford, and that's where that hypertrophic cardiomyopathy paper came from. That was, I guess, one of your lead author papers published in 2020 in, what was it, Physiological Genomics?
Yes. So there were a couple papers that came out of my PhD. That one was looking at a potential therapeutic for hypertrophic cardiomyopathy, which is a disease that affects about 1 in 500 people, causes an overgrowth of the left ventricle of your heart, which does all of the pumping blood out to the rest of your body. And if that overgrows, which can happen because of genetic reasons, you can have problems with blood flowing everywhere. And it can lead, unfortunately, to sudden death often, because typically there are a few symptoms until you hit sudden death. And that's when you hear those news stories about student athletes, just sort of falling over on a field and having a heart attack, it's often something like HCM. So I was my project was two-pronged. One was to try and make this therapeutic, which, we learned some things. The other side of it was doing RNA sequencing to get some better diagnostics. And it started as a side project and then grew into something much bigger and ended up being a really, really fun thing to work on and something that I was really proud of. So lots of time in the lab.
Yeah. Well, yeah, that's what you need to do to get a PhD in genetics. But then you stopped doing that. And now you're doing what you're doing now, which is a lot of things. So you're a freelance writer. You're the founder and I guess owner of Helicase Media. You do the chemistry shorts, which is, I suppose, funded by the Dreyfus Foundation. Everyone here should check those out. The chemistry shorts are wonderful. But all of this is all good. But the audience of this pod tends to also traverse the wasteland and happy land of TikTok.
Yes.
Now, so you got to tell us a little bit about this, what you're doing on the social media side of things. It's TikTok, YouTube, Instagram. What are you doing?
Yeah, so I have been on YouTube now since 2012. So I almost didn't start a YouTube channel because SciShow had just started the year before. And I was like, well, they don't need two science YouTube channels. Like, that's too many, which now we know is absolutely inaccurate. There's a whole ecosystem of science on social media. But I've been doing science on social media for a long time. And it started just because I liked talking about science, and this was a convenient way for me to do it. And now I'm lucky that it's a big part of my job to talk with scientists and put what I've found out there on the internet. And I resisted TikTok for a long time. But at the beginning of the pandemic around 2020, they were doing a big education push, and they asked me to join as part of this education push. And they were like, hey, if you can make 40 videos in 2 months, we'll pay you. And I was like, well, what else am I doing? It's 2020. I'll do it. And I really came to love the interactivity of TikTok. And now I think Instagram has grabbed some of this too, that I can post a 60-second video and people can respond to it, and I can have a conversation with them almost instantly.
Yeah.
And that has been such a fun way to do science communication in what feels like much more real time than YouTube, where I spend, months making a video and then it goes up and then I get 5 comments or whatever.
So getting back to your comment earlier about engaging with the anti-vax inclined population, I have a best friend who does this a lot. And I'm like, why are you wasting your time? Like, you're never going to get through to them. Do you find that it is possible to reach people that are so extremely skeptical of science as a concept?
Yes. And it's great, but also frustrating because it's not scalable. I can spend 90 minutes going back and forth with someone in the comments of a TikTok video. And I've gotten to points where they're like, wow, I didn't get it before and I get it now. Thank you. That's incredible. I actually screenshotted one of the interactions I had yesterday because I was so proud of it, because I don't think this person ended up agreeing with me at the end. I think we had separate viewpoints on vaccines at the end. But in one of her comments, she said, I appreciate having a civil conversation about this, by the way. Thanks, Alex. And I was like, that's great, right? Like, we weren't just yelling at each other. We were having a conversation. And I don't know that I swayed her. I don't think I did. But at least we had that discussion. And I think I put some of these facts out there, right? And other people who are on that page will see them too. You
did more than that. When you have these conversations, even if people don't necessarily agree with you at the end, you've built the brand. I mean, she's going to be thinking about you next time when she's thinking about science thing. And she's like, you know what? I need to bounce this off someone. Maybe she'll come back. You never know. I don't get to have this as often, because more often than not with coffee, people come in with these preconceived ideas and they're just totally baseless. And then you can really quickly drink the experiment. So you can just show them the result. It's a little difficult with It's like, hey, let's all have a vaccine right now, and let's see if we get autism tomorrow.
Yeah. Yeah. Well,
I'm joking, of course, but that's
yes. Yes. I will say I did so much vaccine communication at the beginning of the pandemic, and so many of those people told me I would be dead in 3 years. And it is everything in me to not go back and find all those comments and be like, hi, I'm still here. The CDC did a terrible job of science communication at the beginning of the pandemic. Like, a truly bad job was done by the CDC.
How would you change it?
I'll tell you, the day that I went to the store and started stocking up on things was not in March. It was in February when I was driving home and I was listening to NPR, and Trump came on and he was like, oh yeah, these things don't spread like that. It's going to be fine. And I immediately was like, yes, they do. Turn the car. We're going to the grocery store and we're stocking up on food, because clearly they're telling people to just ignore what I think we in the science community were pretty already worried about, but we're like, maybe they'll handle it. Maybe it'll be okay.
Living from the scientific side, living through a pandemic is pretty, I mean, you really look back in history, there's not a lot of scientists who've actually got to do that because they don't happen all. I mean, there's, it just didn't happen that frequently. When they happen, they're terrible. And we just lived through one. My goodness, you learn a lot. You learn a lot. If I could delete one thing that happened, I would delete that heat map with the growing red dot of doom. Like where it's saying, oh, we've got outbreaks in Los Angeles, we've got it in New York, we got it in Ohio. And I remember feeling heart palpitations, laying in bed, feeling like this helplessness. Even though I assumed that, a young person, reasonable health, I was going to be okay, I still felt like we're all going to die. It turns out I'm still right. We are all going to die, but it's about
true.
That feeling is just awful. Anyway. All right, we could go on and on and on, but
yeah,
we got to pretend like we're talking about coffee here for a second.
Yeah, we're pretty far off of the coffee at this point.
Well, sort of. So why I thought of you when I was thinking of this paper today is because when you think about poorly communicated scientific things in food in general, one of the things that people point to scare people away is this concept of microplastics. And this paper is published in Food Chemistry last year, titled "Pouring Hot Water Through Drip Bags Releases Thousands of Microplastics into Coffee." I think that's a pretty spicy title for I'd say jalapeno at best spicy salsa. Okay.
Yeah.
Now I'm going to ask you, because you're going to know a heck of a lot more than Doran and I about microplastics. Should I be worried about anyone worry about these? What are these things?
Worry is a relative term. So microplastics, the idea is that plastics, we'll start here. Plastics are great, but we use too many of them, right? We're using plastics for everything. And so many single-use plastics, even non-single-use plastics, just get thrown away after a week, a day, whatever you're using them for. And they wind up in the environment and they start to degrade. And when you see like a plastic bottle on the sidewalk, that's pretty easy to pick up and throw away, recycle, whatever it is. But they degrade down into these microplastics that are on the micrometer scale, or sometimes there's nanoplastics on the nanometer scale. And these are small, teeny tiny microscopic pieces of plastic that have sloughed off, broken down from UV degradation, and are now just out there in the environment. And we've found them pretty much everywhere now. They have been found in Antarctica. They have been found on Mount Everest. Like, you think of these remote places, it doesn't matter. There's microplastics there. They're in the air. They're in the water. They're in our food. They're in us. Yeah. So there have been increasing studies that have come out that they find them in blood samples. There was a kind of wild study that came out earlier this year that in 100% of tested placental samples, and this was 100% of maybe 18 or something, 100% of them had microplastics in them. Like, all of these samples from the human body now have microplastics. So we're finding these things everywhere, and we kind of don't know what they do to us yet. They've really only existed on the planet since plastics have been around, which commercially is, I don't know, depending on where you want to draw it, the 40s, 50s, depending on what you're sort of talking about in terms of plastic. But they haven't been around for too long. And now these microplastics are accumulating everywhere. And we have seen in some animal studies, some cell model studies, that they can cause problems. So I think some of the first ones that came out, we were seeing like intestinal blockages in fish. That was one of the first times that people got really worried that they were eating all these microplastics. But now we're finding that they can induce inflammation in animal models. They can induce immune responses. They can mess with the microbiome. But there isn't good data yet on what they do in humans. So we're looking at these things. We're saying, hey, they're kind of everywhere. They're seem to not be so great when we look at them in animal models and in cellular models. And they can carry other chemicals with them. So they can carry along with them thermostabilizers and dyes and additives to these plastics. But we're just not sure. So it's one of those things where there has not been a definitive study that say microplastics are super harmful for you. But the data's not saying that they're likely great for us.
Yeah, I think even in the complete absence of any data, if you just imagine yourself as a person and ask, would I rather have more or less microplastics in my body? You don't need to know anything to say less, right?
Yeah.
But it's, to the point of the FDA, though, they don't have rules set in place for microplastics. And they say it's because there's no standard in detection, quantification, or characterization of these things.
which is this
Yes.
whole paper.
Yeah.
Yes. Yes, it is.
Yeah, so this paper attempts to make some sort of detection, quantification, and characterization of microplastics that are coming out of these single-use pour-over bags that I'll show a picture on in a minute for you, Alex, and we'll post a photo of one on Instagram. This is an interesting thing because in coffee, this is probably not the main source of exposure to plastic. You know, on the inside of most coffee bags, there's going to be some layer of plastic lining. There'll be a one-way valve to let gases out, and so there's going to be contact with plastic there. Basically anytime anything touches plastic, you can get some sort of microplastic coming off of it. In my research in the lead up to this, I was looking at things that have a lot of microplastics, and it's very often it's breast milk, and it's coming from the teat that a baby sucks on, as well as the woman channels microplastics into their breasts. So this is a complex. It's hard to avoid. This is the point. So when we're thinking about having coffee, exposed to plastic, the particular type of brewing apparatus we're talking about today is just one of many ways that microplastics could make their way into the coffee. Okay, before we do that though, I want to talk about the experimental setup for a second. We read through the paper. This paper is not the smoothest read. There are a few curveball sentences in there. One of my favorite sentences of all time in any scientific article. Did you pick this one up, Alex? That the authors write, nowadays it is convenient for white collar workers to order takeout.
I did notice that because they were talking about the takeout containers. And so actually, it's funny you mentioned that because that line made me go and look at where this paper was, where the study is from. And the study was from a research institute in China. And so because I was like, everybody does takeout. And it did make me wonder, actually, if that is a cultural difference between the US and China, that it is a different sort of socioeconomic thing to do takeout. I had no idea, but I agree. That made me wonder, wait, where is this? That's a socioeconomic divider potentially.
Yeah, it's from the National Engineering Research Center of Seafood.
This is a seafood joint that is talking about brewing single serve coffee. Anyway, that they're a single serve. You tear them open. The coffee's already in the sachet. The sachet has arms that reach over the cup. You then pour the water over the coffee. It's like a pour over, but the bag steeps as well. So there's a prolonged contact time between the hot water, which you're going to drink, and the bag. It's really important to note that the way they designed this experiment was they first opened the sachet and they removed all the coffee.
Which was so upsetting to me.
Yeah, I know.
Because that's not how you're going to actually drink this.
No.
Yeah, it's quite interesting because whenever they eventually get to their detection method, they're just doing FTIR, and I can't imagine that the coffee would interfere with it too much.
No. And part of their argument in here was that the drip bag itself was equivalent to a small filter device, which could keep some non-edible substances, including microplastics, in the drip bag. But they don't actually prove that. They just say, well, it would filter them out. And that, if they wanted to do both, I would have been fine. But it was frustrating to me that they only did the scenario that does not match how people would actually be using the product. That was frustrating.
I agree with you, Alex. And we're so, as they continue to tell you what they did, they noted that some of the polyester was seemingly degraded because you brewed it at 95 Celsius. Polyester degrades through base hydrolysis, and coffee is acidic. So it's very unlikely that you would ever actually see this polyester degrade if there was coffee in the bag, because the pH would be sitting at about 4.8 rather than sitting at neutral. So some of the results here, you just need to remember that they were performed using hot water contacting a plastic. So they're trying to incentivize the extraction of microplastics, not the extraction of coffee.
Yeah.
Yeah. Okay. You're frustrated.
No,
all right.
I found it frustrating, and I wish they had done both. I think this would have been a great control if they had done both. But they also make some weird statements in here too. They say, in our daily life, the friction between plastic tableware and the outer surface of the drip bags is consistent with the flushing of water used in our experiments, and then don't prove that at all. What do you mean they're the same? You have to prove to me that they're the same if you're going to say that.
I've been trained to write papers such that in the data you cannot be wrong. You cannot write anything that can be wrong in the results section. And so there's a lot of statements in here that are just kind of tacked on that seem kind of, arbitrary and baseless, but they do reinforce their point. So I think that's why they put them in there, but there's no way that's not part of the study.
And I'm not even saying that they are wrong. I'm saying they haven't proven to me that they're not.
Yeah.
And like that's their burden in this paper to do.
My big issue with the experimental section is they steep this plastic and then they filter it through a metal filter that has a pore size of 10 micrometers, which first of all, I mean, depending on the aspect ratio of the particles that you're getting out, some really big plastic particles might pass through there. Like they show a scanning electron microscope image of a rayon fiber, and you can easily imagine that just slipping through a mesh filter.
Sure, like a small hole.
And
Yep.
the other issue after that is that they take this solution and they sonicate it for an hour. And
Yeah.
if you've never encountered a sonicator in your life before, these things produce a lot of energy. They're making basically little vacuum bubbles in water which rapidly collapse. You get local heating into thousands of Kelvin in some cases. There's a lot of energy in a sonicator. Like we use sonicators to exfoliate materials to peel layers off. And so I cannot imagine that sonicating this solution for an hour isn't dramatically affecting the morphology, the size, the shape, all of the plastics that are in the sample.
Yeah, they just say, "Hey, someone else has done it. Cite them," and that's it. Well, anyway, okay. Those problems aside, there's other things to think about. So usually with things that go in your body that are bad for you, you report them in mass that you've ingested divided by mass of you. So, a big person can typically withstand a little more than a little person can. And this paper doesn't do that. So this paper just counts. And counting is a little bit difficult because if you swallow a toy soldier that is 2 inches tall, that would be counted as one microplastic. Well, that's a macroplastic, but you get the point.
Yeah.
That's one plastic. And so they're basically just going ahead and counting them. And in all the different coffee bags that they sampled, I think there was 8 different bags they played with here, they all released different microplastics. The thing is that these bags and most plastics like this are actually a blend of a lot of different types of plastics. I'm not really familiar with polymer manufacturing and blending. This is sort of outside of my zone.
This was actually the basis of a pharmaceutical company that I worked at.
Oh, is that right?
The whole technology was based on using techniques that are done in plastics recycling and plastics manufacturing to immerse drug molecules inside of a polymer matrix.
Okay, so seeing polyethylene, polyester, polypropylene, and rayon all together is just a common thing.
Yeah, basically polymers typically have a glass transition temperature, and if you heat them above their glass transition temperature, you get them kind of rubbery and things can float around and move around. So if you have enough heat in a mixture of a polymer, you just form a molten thing that you can congeal.
I see.
And so you can get different polymer molecules to interact with each other and make kind of like a big aggregate.
Well, I think the important point here is that each of those, let's just assume that they make this aggregate, they're heat stable at least up to 100 Celsius so that you can brew coffee in it. The polymers are made of different things. Like polyethylene is just carbon and hydrogen. Rayon has a lot of oxygen in it. And so they have different vibrational signatures. And so that's what they're monitoring is they basically have a microscope that finds these plastics and then they look at their vibrational signature and that tells you how much of it is polyethylene, how much of it is polyester and rayon.
Yeah, this part of the paper I think is quite well done. I mean, you can see in figure 3 that the spectra for each of the distinct polymers is quite different and that they're getting pretty good agreement to their reference spectra in the actual samples. So that I think is pretty hard to argue against.
So at this point, what do we conclude, Alex? I think they say at this point, we're just saying there are microplastics that come off of this plastic bag that we're using to brew coffee.
Yeah, and they were able to detect which types in which bags. So there was, bags G and H were only, I think, PET and rayon or something. Yeah, we're only PET and rayon, and the other ones were a mixture of all 4. I think this, for me, is one of the places where I think microplastics research broadly struggles because a lot of the people who are doing microplastics research they have to buy their microplastics from somewhere so that they can have like a nice distributed sample. So a lot of microplastics research is actually done on polystyrene,
Mm.
and it's typically done on like micro-sized polystyrene balls. And so they're spheres, so they don't have edges, right? And they're all of the same size and they're all one material. Whereas I feel like this is a really good demonstration that the microplastics we're actually encountering in our lives are not that. They are mixtures of materials. They are often, if you look at these pictures, sort of jagged shapes because they're just happening from this natural breakdown of these materials or this breakdown of the materials over time. So I think I agree, this is one of those ways it's kind of nice to highlight why this research is hard to do, because how do you standardize something that by its nature is non-standard? That's pretty tough to test.
Yeah, something about the paper that's a little bit opaque is the exact method that they use to arrive at their number for the microplastics. I'm not sure if they looked at an average of these small microscope slides and then extrapolated, or if they backed out some kind of concentration based on this number. And if so, it would have been cool to see if they could reproduce that with some kind of standard. So that part is a little bit opaque to me, so it makes it slightly hard to feel certain about the number calculation. I mean, obviously
Yeah.
the identity of the microplastics is established, the presence is established, but the counting seems like a little bit of a stretch to feel super confident about.
Yeah.
Yeah, you're right. Because in there, I had just looked at like, and I was like, oh yeah, well, they did the counting in ImageJ kind of thing. But they do say the particles with high response values were selected and counted. Like they're doing like a subset counting and then
Yeah.
doing some kind of calculation.
Yeah, and are you counting 8,000? I don't know.
Well, I will tell you the hours and hours I spent in ImageJ counting cells in grad school. I probably did count 8,000 cells. So that's actually not out of the realm of possibility in my brain, but probably not per sample.
The other thing that we should mention here is they steep these bags for 3 different time intervals. And this is important because there's no tricks. If you leave hot water in contact with plastic for longer, you get more of it out. And it seems like the composition more or less stays relatively the same. You just get more of it. And so there's maybe something from a brewing side, if you're concerned about microplastics, maybe something to consider here is find a way to brew quickly, I guess. Yeah, look, there's not a lot to say on that. I think what is kind of interesting is they do a principal component analysis. This is Doran's favorite topic, so I'm going to let Doran just hit it.
Everyone in, I think, any food journal is required by law to do a PCA.
Yeah. Well, this one actually isn't even that
Yeah.
bad. I mean, this one's okay.
Yeah, this one actually looks good. So, principal component analysis, we've talked about it on the podcast before, but basically you're just inputting all of your measured things against all of the variables that you collected. So in this case, it would be all of the different bags, all of the different steeping times, all of the different compositions of the bags, and you come up with some axis that describes the variance in your data. And this axis is a linear combination of weights of all of your variables that you input. And so you can see that on figure 6a, they get a kind of cluster. And they're describing that the clustering, you have one group which is predominantly polypropylene, polyethylene, and one cluster which is predominantly PET and rayon. So that makes sense, I think. The bags are dissimilarly composed.
And I got excited about this because I was like, one of the things that you could do is you could almost like a analytical chemistry lab. You could basically say, can we determine which of these 2 miscellaneous coffee extracts came from which bag? Or, it's kind of cool. And whether it's useful is a different thing, but it is nice that there's clustering. I thought that was kind of neat.
I'm glad that you found a positive in that, because I feel like the data in figure 4, the data in figure 6, it's all the same stuff just shown in different ways. Like
Yeah.
we got bar graphs, we got these swoopy graphs, we got PCAs, we got heat maps. And I was like, you're showing me the same thing in 4 different ways.
Yeah.
I was a little frustrated by that, but you're right. The PCA is maybe the most helpful of them.
Yeah. Actually, as far as food chemistry articles go, I don't mean the journal Food Chemistry, just in general, just work in food. This one, I'll applaud them for one thing. This is one of the very few papers where they try and address only one exact thing, and they do experiments and then write it is one point. And the point being, there are microplastics that come out of these single-serve brew bags, and I guess we'll talk about how much in a minute, but there are microplastics come out, and depending on which bag you use, you get different microplastics based on the different compositions of the bag.
So one figure that I really would have liked to see in this paper is, as we mentioned before, I mean, everything in our modern world comes in contact with plastic at some point or another. Everything. And they have a paragraph in here, paragraph 2.6, where they're basically saying, "No, trust me, bro. There's no, we didn't put other plastic in the stuff." But I mean, you have to imagine if you're using even really pure ethanol, that ethanol has probably been in a plastic container at some point. I would have really liked to see an analysis of just a blank. So just ultra pure water, sonicate it, pass it through a metal filter, sonicate that in ethanol, dry it down to, basically do every single thing without the plastic bag with all the reagents that they used. How many microplastics are in there? What is their composition? I'm almost positive that they would have found some. And it'd be curious to see how much smaller that number is than the plastic bag number.
For sure. And I think that is a kind of necessary and missing control for this study. And I'm going to posit one more thing I wish they'd done that would have made it a different study entirely. But, I don't use this kind of coffee brewing system, but I do drink a lot of tea that comes in tea bags. And there was this whole same discussion around like plastic tea bags a couple years ago, sort of thing, releasing microplastics. And so I would love to see, like, do any of these kinds of coffees come in a paper bag similar to like a paper tea bag? And what are the microplastics out of that? Because I think that also would have been, again, it's sort of a different study at that point, but would have been a control in a different manner of like, this is going through a similar packaging, similar shipping, similar coffee filling sort of thing. Is it better to use one that's not plastic than plastic if you're trying to avoid microplastics?
Yeah, and I mean, cellulose is a polymer. So if you have microcellulose in your beverage, is that going to have the same health impact as a microplastic? I mean, they're both polymeric materials.
Yeah, this goes back to a point that you both were talking about earlier, which is that if you're going to write a paper that people are going to read, like this one, you do, or you should have an obligation to make sure you cover all of the angles on that paper that really hammer home the point. Brewing, for example, in paper, and also doing the control and being really thorough. I don't blame the authors for that. I don't blame anyone for that. It's just that's what you need to do to be really thorough. The reviewers probably should have helped out a little bit. They probably should have asked for that. I mean, that's what peer review is actually for, is like, hey, please present the baseline so that we can actually see that. That's what reviews are supposed to be, Anyway.
Yeah, this is a big science communication point, actually, because I've discovered, I mean, I've been in science so long, I don't even think of this, but a lot of people think that what peer review is that another lab does the experiment and verifies it. And that is completely not what peer review is. I mean, Chris, you can say more about what it actually is.
Yeah, what it is slowing down my time to publication. No, what peer review actually is. I want to hear, Alex, I want to hear your point on it, because you have to tell people about this often too when you're trying to convince them that more likely than not the findings are, have X or Y impact. But for me, what peer review is I write the best article I can and think about everything I can. And at some point, I've read my own work and I've thought about it so much, I can't do more on it that makes that paper more robust. So I send it in, and then the reviewers come back and say, here are a couple of ideas that would make it more robust. That's what it's supposed to function as. There's another layer to it, which is not really important, which is, does the article fit the scope of the journal? Let's ignore that, because let's assume you're sending it to the right place and it's of the appropriate impact. The reviewers are supposed to make sure that everything that's in the paper is in principle reproducible. In our field in computational chemistry, sometimes you get reviewers reproducing the calculations. So I've had that probably 7 times out of 120 something times. And I can tell you that in those 7 times, those were the best peer reviews we've ever got, because they did the job. But we don't get paid to peer review. We don't get any notoriety for it. We get nothing. It's the silent service that scientists serve. And if you do the math, if I publish one paper and it gets 3 reviewers, then I should be reviewing 3 papers for every paper I publish. and there is no way that people are doing that much reviewing. Alex, what do you say to people when they talk about peer review? How do you talk about it?
Nobody talks to me about peer review. Nobody
Really?
outside of science. Yeah, no. You were like, well, when people think about peer review, nobody thinks about peer review outside of science because I think they're just not. And this is a failure of science communication. They don't understand that that's a part of how it works. And I think too often it is talked about that like science is just a monolith, that like a study was done and it found data and that data was correct. And that data just like stands alone. I truly do not think that there is public understanding beyond just the fact that maybe scientists talk to each other. I really think that that's about where it ends, that I think people know that scientists talk to each other and have fields. People broadly do not even know that the peer review process exists. And when people think about editors, I think they think much more of like a newspaper editor who's just like going in and like, well, I'm going to expose how little I know about newspaper editors, but who's doing like story structure stuff and, editing for kind of that second thing of content versus the actual data quality. I do not think people understand that there's a peer review process.
Isn't it? It's an interesting thing because let's go back to the pandemic for a second. It took a while for the first articles in Science and Nature to show up, right? And everyone's sitting on the edge of their seat, the scientific community, but also the media. Everyone was waiting to hear about whether it's, what are we going to do? And we're looking to the scientists to tell us. And it took ages for those papers to come out. And the reason is because you better be damn sure that you've got good data, and then it's got to go to the reviewers, and those reviewers are going to be really, really hard on those papers because they know it's really important. And the papers that did come out are really robust anyway.
Yes, but
yeah,
the other side of that is something like bioRxiv and metaRxiv, which I think are wonderful resources where people can put pre-peer-reviewed research out online. And because people did not understand the difference between peer-reviewed and non-peer-reviewed research, that kind of stuff, which anyone can put something on bioRxiv, basically, there's very little review. And I think that's good. Like, there's a lot of great things that does for the public, but people not understanding what those places were for, just saw a science article, thought it had gone through everything that a normal science article would go through, and took that as the same level of scientific certainty that those other articles would have gone through. And that was a big problem. There was a huge problem in misinformation spreading from preprints, and that stuff is hard to get back. And I think, again, that is a failure from us as a scientific community to tell people anything about how scientific articles get published, because I don't fault anyone who saw an article on bioRxiv that looks just like a normal article and didn't understand why it wasn't and what was different.
Yeah, the context I've come across this in is when, there are these big scandals about data falsification and things like that. Like there was the recent Alzheimer's stuff where it turned out that this one lab was falsifying a lot of images. And people are like, wait, but they were peer reviewed. So didn't someone else have to do it?
No. And I think that's the thing too, that I think that's a very good point, that I think when people do hear that there's peer review and understand what it is, I think they misunderstand it, that they're looking to see if people falsified data or misrepresented things. I think that's what people think it is. And that certainly is some component of it. If you're reviewing a paper and you think there's falsified data, you should absolutely wave that flag. But that's not the point of peer review. And so it's not what people spend their limited time on. They have to assume going in that people are presenting this data in good faith and that it's real data and review it from there. And so it's often afterwards that, after this stuff has been published and now it's been seen by thousands of people, that maybe one of those people sees a weird, funky little edge in a Western blot and then investigates it, kind of thing. Because that's not the point of peer review, is to catch those things.
Yeah. I mean, and the other example is LK99. Just 2 papers show up on the archive and the whole world explodes because I think we got a room temperature ambient pressure superconductor, and of course we do not. Anyway, we have strayed a long way. Let me say, let's get us back on track for a sec. We're not criticizing peer review of this article.
No.
Not at all. Not even criticizing the authors of the article. Of course, we can all improve. What we are looking at here, when you write about something that is a hot topic like microplastics, you do have an obligation to do your very best to make sure you don't mislead the most trained and the least trained readers of that article. And I'm not sure that they achieved both of those goals, and that's okay. That's what science does. Sometimes we need to keep trying and hit that goal. The final thing I want to say on this paper is I mentioned earlier they count the number of microplastics, but they don't weigh them. That's a problem because you need to know how many microplastics and how much they weigh to understand how much you've actually ingested. There's no indication of that in this paper at all. So I actually don't have an ability to make a statement about this, but they do have some funny sentences in the end of the paper saying, according to previous research, humans drink 3 to 4 cups of coffee with an advantage to their health.
Yes, I'm pretty sure I highlighted that exact sentence because I was like, what does that mean?
Yeah, no time frame given.
No.
Yeah, so then they try and make an estimate. They're saying that if you drank, how much was it, Doran? Like 3 to 4 cups a day for your whole life, that you end up with something crazy like millions of microplastics per year in your body that you've been ingesting through coffee alone, brewed this way.
Yeah.
I will say nothing more than the following statement. If you are choosing to drink 3 to 4 cups of coffee per day and you're electing to use the single serve pre-ground in the plastic bag brew method that we're talking about today, you are probably not being the most efficient with your money, because there are a lot of other brew methods where you get a much higher quality coffee coming out of it, where you grind your own coffee and then you use some other brew method.
The white collar worker needs the convenience.
They could probably also order a coffee from
Yeah, at that point, just get yourself a drip coffee maker and just go at it. Like, what's going on?
Yeah. Have we said it all, Alex? I think we may have said it all.
I think so. I think it was an interesting paper. I'm glad that there are studies like this being done looking at these kinds of things, because we don't know, right? I would love a world in which microplastics are having absolutely no health effects on us. And we're like, wow, we kind of wasted our time looking at that, but we learned something. But looking at the data, have a suspicion we'll want to know more where these microplastics are coming from and potentially just reduce their sources in the future. So I thought it was an interesting result. I just wish this feels like figure one, and I want the rest of the paper.
I'm really grateful for that. You come back anytime. Anytime you want.
Please have me back. This was fun. I don't get to dive into papers like this often. Oh, yeah. Ooh, yeah. Let me find a coffee paper. I would love to pitch it back at you.
Not even coffee paper. You can pick any paper you want. This podcast is open for you.
Well, thank you. No, that's very kind of you. And this was super fun. I don't get the chance to really dive into papers like this with anybody anymore. So I miss Journal Club, and I'm happy to have had a little weekly Journal Club. So thank you so much for having me.
Alex, tell us where people can find you on all the different handles.
Yes, I am at Alex Dainis everywhere. It's Dainis, and I'm one of the only, there are 3 Alex Dainises in the world, but the other 2 don't have a social media profile. So if you find it, it's me.
Good.